So this is what I'm doing right now. I have fed sorbitol in different doses to my flies for 2 weeks. And now it's time for me to put them back to sleep so we can extract their RNA through a process called PCR. And then we're going to be looking at degeneration, seeing if there is degeneration of their RNA, of their cells, etc., etc. So this is pretty interesting. Now that I have been working with sorbitol, I see it all over the place in gum and candies and things like that. So this is going to be super interesting, and I guess I'll let you watch me work for a little bit. Now this is for my people who want to see what I am now anesthetizing these flies, and there's been 2 weeks of feeding them sorbitol at different levels, different amounts, doses, whatever. And we're going to see exactly what's happening to their RNA, to their genes, to their cells. I'm going to look at a cell called a gene called the Reaper gene. Basically, it tells a cell to die. It causes what's called apoptosis, or it makes it happen, basically. And so we're going to see if sorbitol does that, because high levels of sorbitol cause oxidative stress. Basically what that means is stress in the cell, which basically tells the cell to die. So we are seeing if that's happening, and so I'm taking the vials, anesthetizing the flies, and then what we're doing after that is putting the flies in the smaller vials, and then I am going to freeze them, and then we're going to break them down. Should be kind of fun, kind of cool. So take these little things, put the fly nap on them like such, and we're going to do 4 at a time to speed up this process significantly. So that's 1, bang, put that in there. Poor guys, only 2 weeks to live. There's 2, put that in there. Flying around like crazy, but that's good. I thought I actually overdid it. Oh. There's 3. There's 4. Alright. So this is control 4 now. And I'll do this one full process here. Sorry, it's kind of annoying as I use my fingers to pick up these little fruit flies, but it's okay. So that's 3, 4, 6, and now I have 5 little flies in there. I have this little tiny vial, which is what they will be frozen in. So put this vial here. Problem is they get staticky and they start bouncing around, which is crazy. And I fold that, put that on top of there, like such, and then move them, plate them into the vial. 1, 2, 3, 4. 5. Yeah. That is control 3. And that's all I got. And this is what I'm going to be doing for the next however long, an hour or so, and I will keep you updated. Thanks. So next, I'm putting the flies, which are being frozen there, or chilled into here. From here, I'll be putting them in the negative 80 degrees Celsius freezer so that we can obviously freeze them and then extract their RNA soon, or their DNA, make it into RNA. So it's gonna be awesome.